My name is Daniel Bohler, and our project was how gene expression in Shigella flexneri may be linked to leaky gut syndrome in the brain-gut connection. Like Dr. Scott said, this was under the mentorship of Dr. Kingsley Dunkley. I'd just like to preface this with a hymn that comes to my mind. All creatures of our God and King, lift up your voice and with us sing, Alleluia, Alleluia. And everything from an elephant to the smallest bacteria declares our Maker's praise. So for a little bit of background, Shigella is a non-motile, so unlike Salmonella, it has no flagella. And it's gram-negative bacteria, like a lot of the enteric bacteria are. These microorganisms, they're facultatively anaerobic, which means if there's oxygen present, they can use that oxygen. If there is no oxygen present, they don't need it. And they're from the family Enterobacteriaceae. Shigella flexneri causes a disease known as shigellosis. It's pretty similar to salmonellosis. There can be varying degrees of severity depending on the species. Some cause dysentery, some cause mild diarrhea. So there's a wide range. Shigella flexneri is a little bit more mild in areas with good health care, and that's going to be important because shigellosis is a diarrheal disease that affects developing countries specifically. And so in countries with malnutrition, poor health care, and infected waterways, when you get a diarrheal disease and you're already malnourished, even if it is not severe diarrhea, especially in young children, you've got a risk of it being lethal just because of the circumstances. And this can be transmitted through the fecal-oral route, so eating with unwashed hands, things like that, and some other modes of transmission as well. So to continue just some background information, so this diagram here shows on the left side, the list shows some effects of gut dysbiosis, which is where the microbiota in the gut get off, which would be like a gut infection. And then some of the inflammatory bacteria that are upregulated and anti-inflammatory bacteria that are downregulated during gut dysbiosis. And if you have inflammation in the gut, and this is chronic inflammation, there's a chance that this can enter your bloodstream and go to the brain, causing neuroinflammation, which is seen to lead to neurodegenerative disease, like Alzheimer's disease. And so that's why you've got the arrows here showing to Alzheimer's disease, because you have increased bacterial amyloids and lipopolysaccharide production, and you've got that inflammation that's now traveling. And it's seen that colonic microbacteria is actually different in patients with Alzheimer's disease than other patients. So there is a substantial difference there. Now, as far as Shigella flexneri specifically, there's some virulence regulatory genes that we wanted to target. One of these, VIR-F, that's the major activator of virulence. It's a transcriptional activator. PFKA is going to activate the first committed step in bacterial glycolysis, so very important for growth and proliferation. CSRA activates glycolysis and represses gluconeogenesis and the citric acid cycle. GAD-B and GAD-C are both a little different, and we're going to get to those later down the line, but they are very important for Shigella proliferation. And so for infection, these genes have to be regulated very, very closely by the bacteria. So we had three very specific objectives. We wanted to establish the growth phase of Shigella flexneri in vitro, so in a test tube, in lysogeny broth. And we wanted to measure the glucose consumption over time by Shigella flexneri in a 0.3% lysogeny broth. And we then, based on those same time periods, we also wanted to determine the relative expression of the target genes that I just named, and which also are glucose metabolizing genes. So as the bacteria is able to metabolize glucose, it's also able to increase its virulence. So for our materials and methods, the first thing we had to do was prepare the bacterial strain in the media. Lysogeny broth was mixed, and it was placed in triplicate tubes, so 3 tubes for each time period, 0, 2, 4, 6, 8, and 10 hours. So we had a total of 18 tubes. We added a concentration of 0.3% glucose with 100 microliters of the Shigella flexneri to each one of those 18 tubes. These tubes were vortexed, and all these tubes were incubated at 37 degrees Celsius. And so for each of these time periods, we measured the triplicate tubes for using optical density. And so that showed using absorbance, you've got a growth curve. It shows how many bacteria are present. After filter sterilization, the samples were also evaluated for glucose consumption, which I'll get to next. And we took an aliquot off the top of each triplicate sample, preserved it in RNA later, so we could check the mRNA expression in the sample. So for glucose analysis, we used a Fujifilm glucose assay kit. You can see the 96-well microtiter plate in the image. That was just to create a standard curve. The glucose solution was diluted down from 500 milliliters, 500 microliter per deciliter down to 31.25. That allowed us to create a standard curve to compare glucose consumption with. And then a graph was constructed, which we'll see later. As far as gene expression analysis, after the RNA was preserved in the RNA later, we extracted that RNA with an RNA purification kit. Primers had been designed from GenBank for each of those genes we wanted to look at, and then we ran qPCR, which amplifies that DNA. It gives us more copies of the DNA and then shows us what DNA is present, how much is present in the sample, so we could get an idea of what genes were being up or down regulated over time. And that's the qPCR machine in the lab that was used. And so here's some of our results, starting with glucose analysis. On the left side, this is the actual growth curve, the optical density that I mentioned earlier. You can see from about 0 to 1 hour, you've got the lag phase with no growth. And then when you start at about 1 to, say, 6 hours, you've got exponential growth or log phase. Then you get a stationary phase and then a declining phase as you head toward 10 hours. Now, if you look at figure 5, the graph on the right, that shows the utilization of glucose by Shigella. And as you look after 3 hours, that glucose utilization almost perfectly follows the growth curve. And so that shows that as Shigella is growing exponentially, you also have an exponential use of glucose, showing that it's using the glucose in the meat to proliferate, which is a good sign that we did something right. These are the results for the gene expressions for 3 of the genes, VIR-F, CSRA, PFKA. And so on the left side, the first bar for each of those graphs is 16S rRNA. That was our control. That was the gene that we expected to be expressed consistently over each of the triplicate samples so that we knew, okay, this is how much we can start with to show a relative expression, whether it's up or down regulated. And so we've got 4, 6, and 10 hours for each of these genes, and at 10 hours, all 3 of the genes assayed were upregulated at least 3-fold, which, as each of these genes is important for glucose utilization, as you upregulate these in direct correlation with increased use of glucose, it suggests that these genes are playing a role in glucose consumption. Now, these are for GAD-B and GAD-C. GAD-B encodes a glutamate decarboxylase. So in our intestine, we've got pretty low pH, and you think if something has to be ingested, it has to go through your stomach acid, has to go through the small intestine, has to survive very low pH before it can proliferate. And so what GAD-B does is take glutamate from the extracellular environment, pulls it in, decarboxylates it, and pushes hydrogen ions, puts them on the glutamate to increase the pH of the inside of the cell while the outside of the cell stays at a low pH. GAD-C is just for the antiporter that brings in the glutamate and pushes out the GABA, which is what is made when you decarboxylate the glutamate. And so you can see also that these genes were also upregulated compared to the control at pH 7, which is what our study was in. And at 10 hours, all of these were also upregulated, even though they were upregulated earlier, which does make sense. And also within this same study, this came from a separate study that was done before my study, VIR-F was also upregulated during the exponential phase, which shows results that are consistent with ours. And so for the discussion, Shigella flexneri demonstrated lag phase from about hour 0 to 2, exponential growth from about hours 2 to 6, and stationary phases from 6 to 8, and the declining phase from 8 to 10, which is just a standard bacterial growth curve. The growth of the bacteria during the incubation was directly correlated with the usage of glucose, which is what we would expect to see because they have to have some sort of energy source to proliferate. As far as gene expression, all the genes that we assayed for were upregulated during the growth of Shigella flexneri, which is a good sign because these are supposed to be virulence activators. So as they are upregulated, as the bacteria is growing, that shows that these genes are doing what they're supposed to do and that we measured them correctly. Gene upregulation was correlated with increased utilization of glucose. So the more glucose that was utilized, these genes were upregulated during that exponential phase. So God gave Shigella flexneri genes to utilize glucose under stress, because for a bacteria in the human body is stress. This suggests that these genes are necessary for growth, proliferation, and virulence of Shigella flexneri, and expression of these regulators can lead to something known as leaky gut. When you get an enteric infection, you can get increased colonic permeability. So you have a destruction of your barrier, and things that shouldn't be in your bloodstream can now get into your bloodstream. And so that's what you can see here over on the left side. When you see all the colorful bacteria down there, that's the lumen of the gut. That's where all your food and things are supposed to be. As they produce short-chain fatty acids, these different bacterial metabolites, and as your immune system tries to fight that infection, you get all sort of cytokines, inflammatory molecules, that when you get production of inflammatory molecules and you combine that with a gut that now lets things through, you can get entrance of those inflammatory molecules into systemic circulation, which is shown here on the diagram, or it can actually enter the vagus nerve and climb to your brain that way. When you get those inflammatory molecules to the brain, you can activate astrocytes and microglia in the brain, which lead to the amyloid beta plaques that are deposited in Alzheimer's disease, lead to them being deposited and prevent your body from being able to clear them. These graphs here were from a study where they looked at pro-inflammatory bacteria, and you can see on the x-axis, you've got Escherichia Shigella abundance. And so with increased abundance, these 3 separate inflammatory cytokines all increased. So it's showing that as Shigella increases in abundance, inflammation increases as well. And so we can conclude that Shigella is using glucose to fund the energy for its growth phases as it's upregulated along with the growth curve. Glucose is consumed in correlation with the growth rate after 3 hours. And gene regulation is tied to glucose consumption, and so gene regulation impacts virulence. Because as these genes affect glucose consumption, that's also going to affect whether or not Shigella can grow and proliferate in a certain environment based on whether these genes are present and doing what they're supposed to do. Because all the target genes were upregulated during growth, we can conclude with pretty reasonable certainty that these genes are important for Shigella infection. And if for future directions, if we were able to find some sort of treatment or drug that was able to target these specific genes as opposed to using just a broad spectrum antibiotic or something like that, it would be possible to decrease the rate of infection, to treat infections without destroying all the good bacteria that is your only last line of defense to fight the inflammation you already have going on. So I'd like to thank the STEM grant, the Gail Dillard Foundation grant for funding this research. Thanks to Dr. Dunkley, who has been the mentor and has been fantastic through this project. And to William Garcia, who did actually all the benchtop research for this, who's now at the Dental College of Georgia. So any questions? Not the hands-on. I did a pretty major lit review and tried to come up to date, and then did some lit review on top of what had already been done. All that was here was the gene expression stuff, and I did the brain-gut connection research to try to tie that in with this. So I was just mainly lit review. I feel called to a career in medicine, and so when you connect microbiology and infection with human health, especially in developing countries, in countries who have little access to the resources we have here, I think is very pertinent to medicine and to try to help as many people as possible, not only with Shigella infection, but also with Alzheimer's disease. Because it's been seen that as you treat dysbiosis in the gut, you can actually help some of the consequences of Alzheimer's and even kind of backtrack some of the damage that's already been done. So if we used antibiotics, we wouldn't be able to, most likely. Even though there's some antibiotics that are more specific to certain bacteria than others, the way to avoid killing the good bacteria is if we could target some of these virulence genes that are specific to Shigella or specific to bacteria that infect the large intestine, so that you turn off some of these virulence regulators without killing good gut bacteria.
And I guess that's what I'm missing is how do you do that?
That would take a lot more research to be able to specifically target and turn off a gene. All we know now is that these genes are important and that it should be looked into, but I'm not sure how exactly you go about targeting specific genes.